and now I'm talking about a research.
Recording in progress.
Okay, good morning, students. I'm so sorry because I have a very important work this to finish and make you wait in about 30 minutes. Okay, can you clearly hear my voice?
Yes, teacher.
Okay, thank you very much. So that today now we start. I would like to remind with you, maybe you forget during take a long time the lecture already studied, right? So in final examinations that we have the form is the same with the midterm examination. That means we have about the 60 to 70 the point for the multiple choice questions, and you will have about 30% of the writings. So now first, I would like to start with the first class, the first lecture to you. That we also have the cell physiologist in the final examination. So please to keep in your mind, and we have how many kinds of cell. Basically, you have the 6 kinds of cell, right? Somatic cell, stem, that we have the totipotent stem cell, pluripotent stem cell, and the multipotent stem cell, oligopotent stem cell, union-potent stem cell, and the germ cell. And please to keep the understand about the characteristic of each kind of cells and give the example. For example, if the question is the totipotency, you can understand about the totipotency mean the cell can differentiate and become the full-term development from the human being. That means can development to the baby, right? So, and can you give the one example? For example, zygote, 2-cell embryo, 4-cell embryo, early A-cell embryo, is the cell have the totipotent characteristics, right? And next is the pluripotent characteristic cell. And what kind of cell? Pluripotent stem cell mean the cell have the ability differentiate and become all organ in our body except with the placenta. Okay, except with the placenta. So, and we'll give the example about the pluripotent the stem cell. Is the embryonic stem cell induced pluripotent stem cell, right? So induced pluripotent stem cell is the cell reprogramming from the somatic cell. And then the for potent stem cell is the multipotent stem cell, the oligopotent stem cell, and union potent stem cell, and germ cell, right? Please to keep this in the carefully. And in the cell, we'll go to the also I just make with you what organelle you need to study. What organelle you need to study? The organelle you need to study is the centrosome. The centrosome. because the centrosome very important to establish the spindle during the cell division. So the centrosome, and I just give you the one important proteins in the centrosome is the gamma tubulin, right? And the gamma tubulin will be the recruits and the microtubulin to establish the spindle. First, centrosome. The second you need to study is the very important for the gene expression, protein modification. So please to study what is the structure and the function of the round endoplasmic reticulum system. And also please to study the smooth endoplasmic reticulum system, because the smooth is the how host the calcium. That's very important for example, very important for the cell, the activation of the S during the fertilization, right? You can see the sperm enter to the S, and the key point is the calcium oxalation from the smooth endoplasmic reticulum system. And the other very important of the protein, very important of the calcium is stored in the smooth endoplasmic reticulum system is the involved into the membrane potential. And you already studied about the contractions of the Hertz muscle, and the normal muscle is the different, right? And that's with the needs to the calcium in the Hertz muscle, and for the prolong of the depolarization phase. And the down of the repolarization phase also the very slowly. That's also the key of the induced by the calcium. Okay, please to reach in the the smooth endoplasmic reticulum system. In the cell, I not to show here in in basically the cell, please cell division based on the control of the microfilaments of microactin, and the chromosome, the separate affected by the microtubulin, right? So that's the key for you to understand when you want to keep the chromosome not the extrude to become the daughter cell. So that means we should be inhibit the microactin. But if you want to the chromosome stop at the metaphase, that means you should be treatment with that inhibits the microtubulin, the polymerization, something like this. Right? Because that's many applications. If you understand that cell physiology, you can produce the theraploid embryo. You can calculate the number of the chromosome, and you can apply the many techniques, for example, to induce the parthenogenetic embryo, diploid parthenogenetic embryo, right? So based on that knowledge. The organelles is just about that, and also we're talking about the nucleus. And nucleus have the inside nucleus also have the nucleolus, right? And then the nucleus is how host the DNA. When we're talking about the DNA, we should be to understand about the gene expression, including the 3-step. Basically is the transcription to produce the messenger RNA occur in the nucleus, and then the messenger RNA will move out of the cytoplasm and combine with the ribosome to then the translation occur to produce the peptide, right? And then the peptide will move to the round endoplasmic reticulum system. Okay, blah, blah, blah. Okay, you already understand. Okay, this day I already add with you here. Yeah. The cell should be understand about totipotency, pluripotency, multipotency, oligopotency, union potency, and drum cell. Okay, in the cell membrane, okay, just a study about that. Everything I already talked today, you will have in the final examination, right? And we will move to the okay, can you see the slide about the basis event during the pre-implantation development?
Yes, teacher.
Okay, thank you very much. Yeah. Now we will go to the first the cells, the physiology, and in the cell physiology, they have the very important the one kind of cell we call the embryo, right? The embryo. So please to understand the pre-implantation development of the embryo, that means we will study about the important event occur in the pre-implantation development. The first is okay, look in here. Student, please to understand about the structure of the zygote and please to explain the difference between the female and male in the chromosomes, the morphology, in the histone modification. That means epigenetic difference between the male and female. So you will be talking about the DNA methylation, the histone methylation, histone phosphorylation, and histone acetylation. And the reason why the male pronucleate is a big size more than the female, and the male pronucleate have the high transcription compared with the female. And the pre-implantation development have the many important events occur here. First, zygotic gene activation. The zygotic gene activation is the type for the embryo can produce can transcription and translation to produce the protein by themselves. Okay, and in the mouse, the zygotic gene activation occur at the 2-cell embryo. However, in the human and cow and the pig, the zygotic gene activations are a little bit late, about from 8 cell to 16 cell, right? Okay, that's mean the talking about zygotic gene activation. Now we're talking about the first differentiation of the embryo. The first differentiation of the embryo is the time of the embryo lose the totipotency and transform to the pluripotency. And then that process you affect by the zygotic gene activation. Okay. By zygotic gene activation. So the effect by the pluripotency, the differentiation, and you should be understand about you should be remember about the octophore express. The octophore express from the 2-cell, the 1-cell embryo, 6 hours after sperm activation the S, and then maintain 2-cell, 4-cell, 8-cell, and in the mouse at the late 8-cell embryo, one important gene expressed in the CD is2, and CD is2 gene expressed to make that cell become the trophoblast cell, and the other cell is maintained in the inner cell mast cell, right? That means inner cell mast cell have the OCT4 expressed. And next, the other gene important is the NANOX gene. The NANOX gene expressed in the inner cell mast cell make the cell become epiblast layer. Okay. So, and then after this, you should be understand the STAT3 genes, to make the STAT3 gene, you make the embryo, the cell have the cell renew characteristics, right? And then they become the epiblast. In this, also please to understand about what is the gastrulation. The gastrulation is the process of the epiblast cell line differentiation become the three germ layer, ectoderm, endoderm, and mesoderm. Okay. So that means the plate in the cell, the embryo should be understand about that. Here also the very different, okay, this are you talking about in the human reproductive, reproductive physiology. Okay, this I already explained about, right? So that means in the embryonic stem cell, you should be looking this slide to, or the other slide to explain, this slide to explain, and keep your mind in this slide, you can understand everything, right? Now we move to the other. The membrane potential. Membrane potential is the key point here. You should be understand the different concentration between the sodium and potassium from outside and inside, right? And the exchange of the potassium and sodium starting to make the membrane potential, action membrane potential. And the exchange of the sodium from outside go to the inside, they result into the depolarization, right? And then after that, the movement of the sodium from inside to outside, that result into what? That result into the repolarization. So the exchange of sodium potassium between the membrane to make the membrane change the electrocharge, finally to make the electro signal in the cell membrane, right? In the cell membrane. So please to understand what is the depolarization, repolarization. And also deeply to understand at the end, the connection between the interneuron and the efferent by the synapse, At the synapse store with the neurotransmitter, right? And the neurotransmitter can relieve exocytosis out of the neuron synapse by the stimulation by the enter of the calcium. So that means the calcium is involved into the neurotransmitter exocytosis, right? To relieve in the outside and to connect with the next cell and bound into the neurotransmitter receptor, then open the sodium channel and the action potential starting again. So here membrane potential just needs to understand the mechanism of how the cell, how the neuron cell can send information to the target organ based on the depolarization, repolarization, and hyperpolarization. Okay, and also talking about the neurotransmitter exocytosis affected by the control by the calcium. Okay. And in here, I also add with you the very important is recognize the proteins. Recognize the proteins is that this is the very applications in the biotechnology. You understand the physiology of the recognize the protein have for the T cell recognize the different cell type of the body. So, but I can see the slide. You talk just pluripotency, and then ESL. So slide have the totipotency, right? Pluripotency of ESL, and also the induced pluripotent stem cell, then iPS. iPS cells also the iPS cell also have the pluripotency. Not only the embryonic stem cell, recently the scientists already demonstrated that the induced pluripotent stem cell, that means the cell reprogrammed from the somatic cell to iPS cell can produce that. Okay, Hân have any other?
Dạ thầy ơi, hình như là thầy đang chiếu cái slide của IBS đó thầy, nhưng mà thầy nói tới cái chỗ kia rồi.
Really? Okay, sorry.
Dạ em cảm ơn thầy.
Dạ cảm ơn em. Dạ sorry các bạn ha. Các bạn đã thấy chưa?
Dạ rồi ạ.
Okay. So please to study about the recognize the proteins. Recognize protein is the one is the very important. Recognize protein is that I already used about the. You remember in American they use the horse pick transplant into the human, right? So that means they knock out the recognize the protein, and this recognize proteins is the name. I maybe in the some lecture I talk with you is the 1,3 alpha galactotransferase enzyme. That's the special recognize the protein in the pig, and if you we knock out the recognize the protein, the because can transplant into the human. That's case in the occurs already clinical trials in Americans, but that's the unfortunately the patient die at the 3 months. because that's patient already on, right? Okay. And so for the membrane potentials, that just for membrane, just understand about what is the membrane potential and the action potential, and the calcium, the potassium, sodium difference between the inside the membrane and outer membrane, and the neurotransmitter, the relief exocytosis under effect of the calcium. Okay, here. Anyway, membrane, please to remember this line. What's the molecular can move to the free move to inside and outside of the cell membrane, and what's the molecular cannot enter to the cell membrane? Okay, please to remember this line. Okay. Okay, membrane potential is the just request about that. And the chapter number 7, peripheral nervous system. In peripheral nervous system, please to study and remember about this one. Looking in this slide, and you can please to compare the sympathetic nervous system and the parasympathetic nervous system in the right. And looking in this slide, you have the many questions. For example, the sympathetic nervous system, where they come from? They come from the nearly the central, the spinal cord, right? Lumbar of the thoracic. That means the spinal cord in the center of the spinal cord.
Yeah.
And the other is the parasympathetic nervous system. They come from the cardinal and the sacral. That means in the brain and into the end of the spinal cord, right? And compare the different. What is the long, those pre-colonial fiber, post-colonial fiber, and what's the neurotransmitter relief? That means.
Thầy ơi,
Thầy ơi, tụi em chưa thấy slide.
Yeah, sorry. I also sorry. I open first. I not share. Yes, in the peripheral nervous system, please to keep your mind in this slide. Okay, now you already see, right? Sorry. Okay. Okay, for this chapter just talking about that compare what is the sympathetic nervous system and what is the parasympathetic nervous system, and compare the two systems. Of course, you know the parasympathetic sympathetic nervous system is the active system, and the parasympathetic nervous system is the system to relax the system. Okay, this chapter just concentrates about that. And now we go to the what we use study in the circulation system. In circulation system, students should be okay. In circulation system, the student should be another. What is the components of the circulation system? Heart, blood vessel, and the blood. Okay. And you should be understood the circulation system divided into two systems. One is the pulmonary system, the circulation system, and systematic circulation system. Pulmonary system is the system from the heart to lungs, and the systematic the circulation system is the system from the heart to all parts of our body. For that, and because the heart lungs are very near the heart, so that means no need the heart to more high contractions, but to make the gradients of the pressure gradient. So that means the pressure in the pulmonary system usually lower than the systematic circulation system, right? And from that, you also explain why the rise in the left wall of the heart and right wall of the heart is the thin and taste. Okay, that means that should be the divide the was the circulation system. Some divide into the two systems, pulmonary system and the systematic circulation system here. Okay, in this slide. And finally, the heart, just last one, and then the contraction of the heart. Please to look in this slide. Here,